Welcome back, everyone. So we're going to continue on with Chapter 5, How Cells Obtain Energy. So in the last two videos, we learned about how cells obtain energy through cellular respiration, which they're done in the presence of oxygen. But what happens when oxygen is not present? Cells can continue to obtain energy through a process known as fermentation. So that's what we're going to examine in this video, 5.3, fermentation. So by the end of this video, you should be able to discuss the fundamental difference between anaerobic cellular respiration and fermentation, as well as describe the type of fermentation that readily occurs in animal cells and the conditions that initiate that fermentation. So we talked about the first part of fermentation in detail already, which is that glycolysis part. However, it doesn't just end there. We can't just continue to produce NADH and ATP, because then we'll run out of NADH. And so we have to have a way to strip the NADH electrons from NADH and oxidize it to NAD+. And so that's why we have to undergo fermentation in order to bring the product back to NAD+, to the beginning stage to receive more electrons, so that we can continue doing glucose over and over again. Okay, so we have to regenerate NAD+. And that's why fermentation is different from just doing glycolysis. Okay, so we'll go through that whole process. So we're going to begin with a video, of course, from the Amoeba Sisters on fermentation. So this will be a good starting point. And then once we get that understanding of the two types of fermentation, so they'll go through alcoholic fermentation as well as lactic acid fermentation, then we'll go into more detail.
When I was a kid, swimming was one of my favorite things to do. I was on the swim team starting when I was 4 years old, and then on and off throughout grade school. Although I wasn't especially fast. I just loved the water. I used to dream that there was a way I could be like a fish. When I was little, I had a misconception that fish didn't need oxygen. Later on, I learned that no, most fish have gills that allow them to extract the oxygen, which they need, from the water. So then I just thought it'd be really cool if I had gills. But alas, no gills for Pinky. Oxygen is a really big deal. So many organisms, from fish to plants to humans, need oxygen. And yes, even though plants make oxygen in photosynthesis, they still perform cellular respiration, and therefore plants still need oxygen themselves. There's often a misconception that plants don't need oxygen. That's just not true. So why do these organisms need oxygen? It's similar to why you need oxygen. If you've ever wondered why you need to breathe, which is done by the respiratory system in your body, zoom into the cell level. Cells in your body use the oxygen you inhale to perform cellular respiration. The formula here requires inputs, otherwise known as reactants, to make ATP. And oxygen is one of those reactants in the overall equation that is needed to break glucose down in forming ATP. Why ATP? ATP stands for adenosine triphosphate, and it's action-packed with 3 phosphates. It has the ability to power many cellular processes. Typically, it's coupled to other things that it may be powering. Now, after losing the phosphate, the molecule is ADP, adenosine diphosphate, because it has 2 phosphates. In cellular respiration, though, there are enzymes that can add another phosphate to it and convert it back into ATP again. This particular formula of cellular respiration is aerobic, meaning overall it requires oxygen. It's pretty complex, and we have a video breaking down the steps. But that's not what this video is about. This video is about what happens when there is no oxygen, because cells still need to make their ATP. So what kind of cells can handle the no oxygen thing? Well, many types of bacteria can, many types of archaea, yeast, which is a fungus that could be helpful, like making your bread rise, your muscle cells can, for a while anyway, and these are just a few examples. Now, these organisms handle the lack of oxygen in different ways. Some organisms, such as some types of bacteria or archaea, can do anaerobic respiration. They can continue to perform glycolysis, Krebs, and the electron transport chain, just like aerobic cellular respiration. But since there is no oxygen to be that final electron acceptor at the end of the electron transport chain, they use something else, sulfate, for example. These organisms are specifically adapted to be able to use a different electron acceptor in this anaerobic respiration. Another option is the organism may just stick with doing glycolysis, which doesn't require oxygen, and then the addition of some way to get their NAD plus back. We'll talk about what that means in a minute. This process is called fermentation, and that's what we're going to focus on. Fermentation is a way to be able to handle the little to no oxygen issue. It allows for glycolysis to happen and for glycolysis to keep going. That means making ATP where there is no oxygen. And while you won't make as much ATP in this process as you would aerobic cellular respiration, you can't be too picky when oxygen isn't around. Okay, so recall what glycolysis is from our cellular respiration video. In glycolysis, you take glucose, a sugar, and it gets converted into pyruvate. This takes a little ATP cost to actually start it up, but overall, per glucose, you make 2 net ATP molecules, and you also produce 2 NADH. Recall that NADH is a coenzyme and an electron carrier. We also need to mention that NADH didn't just poof appear as a product. No, because NAD plus actually was reduced to NADH when it gained electrons. And if the words reduced and oxidized are confusing, you can remember the famous Leo Gurr mnemonic. Lose electrons oxidized, gained electrons reduced. So NADH to NAD plus is oxidation because it loses electrons, and NAD plus to NADH is reduction because it gains electrons. Now, NADH, the electron carrier, would normally be delivering the electrons gained to the electron transport chain, if this was aerobic cellular respiration. Once losing their electrons, NADH would be oxidized into NAD+, and be ready to be involved all over again in glycolysis. But there's no electron transport chain in this fermentation process, so we've got to regenerate the NAD+ somehow. NAD+ is needed here, after all, for glycolysis to continue. Fermentation, therefore, adds another little step to the end of glycolysis, a step to help regenerate NAD+. This happens because fermentation allows NADH to give its electrons to an electron acceptor, which, in the 2 fermentation examples we are going to give, will either be a derivative of pyruvate or pyruvate itself. So here we go with 2 types of fermentation, which both result in different products from pyruvate. Alcoholic fermentation is done by some types of yeast. So first glycolysis happens, which yields 2 net ATP, 2 pyruvate, and 2 NADH. Now we need this step to regenerate the NAD+, so we can keep doing glycolysis. The 2-pyruvate is used, which will ultimately produce carbon dioxide and 2-ethanol, alcohol. But the derivative of pyruvate shown here, acetaldehyde, can act as an electron acceptor in this process, so the 2-NADH can be oxidized to 2-NAD+, so that glycolysis can start all over. Since alcohol is a waste product in this process, this is why many yeast are used to produce alcoholic products. Yeast also can do alcoholic fermentation in making bread, and the carbon dioxide product we mentioned is involved with helping the bread rise. The tiny amount of alcohol produced in the short fermenting time of bread will evaporate in the baking process. Lactic acid fermentation is done by cells such as your muscle cells, for example. While your muscle cells can do aerobic cellular respiration, they can shift to lactic acid fermentation if they experience an oxygen debt. This could happen if you're working out very intensely, where your blood is unable to deliver a sufficient amount of oxygen to them for their demand. Just like alcoholic fermentation, we start with glycolysis, that yields the 2 net ATP, 2 pyruvate, and 2 NADH. But now we need the step to regenerate the NAD+, and this step is a bit different from alcoholic fermentation. The 2-pyruvate on the reactant side will ultimately yield 2-lactate. The pyruvate can act as an electron acceptor, allowing NADH to be oxidized to NAD+, so that glycolysis can start all over. By the way, this lactate product, or specifically its other form, lactic acid, has often been blamed for the muscle soreness that occurs after intense exercise. In many of my teaching years, this was often discussed, but there's some recent research that may contradict that. Science is so cool. Check out our further reading suggestions and our video details to learn more. Lactic acid fermentation is also done by bacteria that are involved in making yogurt, and lactic acid can contribute to its sour taste. So overall, fermentation is a pretty remarkable process. Although, it does make us appreciate oxygen because despite how absolutely awesome fermentation may be, it just can't make as much ATP as aerobic cellular respiration. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so that was a good introduction into fermentation. So let's get into the details. So as stated before, in fermentation, the reason why we have to have fermentation is because we need to oxidize NADH to NAD+, okay, so that glycolysis can continue over and over and over again. So so we need to have a final electron acceptor, just like in aerobic respiration. So in aerobic respiration, the final electron acceptor is oxygen. But we don't have, glycolysis, we don't have a final electron acceptor. So we need to undergo further steps in order to have that final electron acceptor. So we're going to talk about the final electron acceptors, which are organic molecules in fermentation. It's an inorganic molecule when we're talking about anaerobic respiration. And I think we'll talk about that in a little bit. But let's focus on fermentation first. And there's two types. So we're going to talk about lactic acid fermentation and alcohol fermentation, both which were discussed in the video. And in the video, talked about lactic acid fermentation is something that we can do in our cells, as well as bacteria. And then alcohol fermentation is something that we can use for baking and for the production of beer and wine and things like that. So we're going to talk about both of these processes. So let's start with lactic acid fermentation. So again, some animals use this, including us. So when we are basically working really, really hard at some sort of exercise, and we simply cannot take in enough oxygen, then our cells will have to switch over into lactic acid fermentation in order to get just enough ATP to continue that activity. Okay, so imagine you're sprinting to the finish line, the 100-meter dash, and you're at the 80-meter line, and basically you've used all your oxygen, you have 20 meters left. So that's when your muscles might have to switch over to this lactic acid fermentation to get you to that finish line. Okay, so it'll give you just enough ATP to get you there. All right. And so also we saw from the video that some bacteria do this. So some of the bacteria that are in some of our dairy products, such as yogurt, that is used to basically make the yogurt taste that kind of sour taste to it. So that breakdown of the sugars in the yogurt produce that through fermentation, produce that lactic acid or lactate. Okay, so what are the steps that are involved? Well, the first step, it's glycolysis, and the whole process of glycolysis. Remember from 2 videos ago, we learned about glycolysis. So there's that energy investment phase, which requires 2 ATP to be used. And then the energy payoff phase, where we actually gain 4 ATP from that process. And we end up with a net gain of 2 ATP total, okay? So we start out with our reactant glucose, okay? So, and then we break that glucose into pyruvate, okay, just like in glycolysis. So at the end of glycolysis, we have 2 pyruvate, we have 2 ATP, and then we have 2 NADH. All right. So if we just stop here, if we just keep doing this over and over and over again, we would run out of NAD+. Okay, you can see here we would just have a buildup of NADH, and then we wouldn't have any NAD+ left in our bodies. And so we have to continue with another step in order to oxidize NADH to get rid of those electrons, to oxidize NADH back to NAD+. So that's why we have this next step. So in this next step, we're going to strip those electrons from NADH to oxidize them to NAD+. And in the meantime, we're going to convert pyruvate to lactate, okay, or lactic acid. Okay, so we're going to basically remove the electrons, we'll add them to lactate. So now we have lactic acid. And so the final product is lactic acid or lactate. All right, so that whole process, we started with glucose, and then we're going to end with 2 lactic acids and 2 ATP. All right, so just enough energy to get us to that finish line. Okay, so certainly not the yield that we would get through aerobic respiration, through cellular respiration, but you know, it can work in a pinch, right? So we can get some energy from that process. The other type of fermentation we're going to talk about is called alcohol fermentation. So alcohol fermentation is used by yeast. Yeast is a type of fungus. It's a single-celled fungus. And just like in lactic acid fermentation, it involves the first step being glycolysis. So again, in glycolysis, we're breaking down glucose into pyruvate. And in doing so, we're reducing NAD+ to NADH. Okay, so now we have this NADH that has these electrons. So instead of oxidizing NADH to NAD and stripping those electrons to lactic acid, those electrons are going to be accepted by ethanol. Okay, so it's a different end result here. Okay, so we end up with ethanol instead, and carbon dioxide is produced. All right, so ethanol is the final electron acceptor, as well as carbon dioxide. And so that's why, you know, if you think about it, if we're making beer, for example, we're putting yeast, that single-celled fungus, in with the hops and barley or whatever we're making. And so the yeast is going to be eating the sugars. And as it's eating the sugars in this sealed environment, there's no oxygen there. It's going to be undergoing glycolysis. It's going to be breaking down the glucose, and it's going to be producing alcohol as well as carbon dioxide, the bubbles, right? So you're going to have bubbles being produced in this sealed environment. And it's going to be, you know, getting ATP from that process to power the little fungus yeast cells. All right. So the final electron acceptor would be ethanol in this case, and then we have oxidized the NADH to NAD+, so that NAD+ can then be used over at the beginning of glycolysis once again. Okay. So that is the process of alcohol fermentation. And as you can guess, you can see that this could also be useful in bread making. So we can add yeast to flour, and then warm water activate the yeast, and then the yeast will start eating the sugars in the flour, and that will cause the dough to rise, right? Because we have the carbon dioxide being released, which causes the dough to rise. The alcohol usually gets burned off when we actually cook the dough to turn into bread, but you know, you end up with the little holes in the bread, which give it the rise of the bread. Right? Okay, so that was fermentation. So fermentation basically is just glycolysis with an extra step to accept the final electron acceptor. And that final electron acceptor is some sort of organic molecule, whether it be lactic acid or if it's ethanol, something like that. In anaerobic cellular respiration, we use an electron acceptor other than oxygen that is inorganic. Okay, so it's going to be, in this case, it's going to go through the same process as cellular respiration, where the cell will go through glycolysis, it will go through the citric acid cycle, and it will go through chemoosmosis. Okay, so it's going to go through the same process and produce similar amounts of ATP as with cellular respiration. Maybe a little less, maybe it might get like 30 ATP instead of 36 on average. But the only difference is, instead of getting oxygen as the final electron acceptor, it's going to have some sort of inorganic molecule as its final electron acceptor. So something might be like a sulfate, or in this case you can think of like a methane, something other than oxygen. Okay, so an example, you have like a methanogen. So methanogens, they reduce carbon dioxide to methane, and so that gets oxidized to NADH. Okay, so they'll go through the same process of cellular respiration as we do. They have, you know, mitochondria and same as we do, but then instead of pumping out, you know, or taking in the oxygen, these methanogens will take in, basically they'll use the methane instead as the final electron acceptor. Okay, and methane, if you recall, is CH4. Sulfate reducing microbes, so sulfate reducing, that would be a sulfate, is going to take in the electrons to form hydrogen sulfide. So that is, again, an inorganic molecule, so that's a different type of anaerobic cellular respiration. Okay, so there's a couple of different other types of cellular respiration that are done without oxygen present. So they're anaerobic. So think anaerobic and without aerobic oxygen. Okay, so this is different from fermentation because we still use chemoosmosis and, you know, the other steps involved. All right. And you can see, like, here's some examples. This is, these are like anaerobic sulfate reducing bacteria that release hydrogen sulfate gas as they decompose in algae. A lot of these you see in areas that are devoid of oxygen, so like deep in like hypoxic zones, which are areas that don't have oxygen present, so like in really like swampy, muddy soils, or you know, deep sea vents, things like that. Okay, so let's talk about some types of anaerobes. So anaerobes, remember, an means without, aerobic means without oxygen. So these are organisms that can survive in environments where oxygen is not present. So a facultative anaerobe, they can switch between aerobic respiration and fermentation depending on the availability of oxygen. So an example of that would be our yeast. Okay, so yeast, they can, you know, do their thing when oxygen is present. They can also do their thing when no oxygen is present. Okay, so they would be an example of facultative anaerobes. Okay, so If they're doing aerobic respiration, they're not going to be producing any alcohol though, right? So we would prefer that they do their thing when there's no oxygen present. An obligate anaerobe is a little different because they live and grow in the absence of molecular oxygen. And they tend to only survive in areas that don't have oxygen. So when they're exposed to oxygen, like oxygen gas, they could end up dying. It could be lethal to them. Okay. So examples of obligate anaerobes are, again, anaerobes that live in environments such as like swampy, boggy areas that are like in mud deposits. Also, again, down in deep sea vents where you have high chemical content but no oxygen. So there's just, if you go to Yellowstone and you have these, hot springs, there might not be very much oxygen present, but you have a lot of different like sulfates and things like that there, so they could continue doing anaerobic respiration. So here's like an example of an anoxic zone, so that's basically low oxygen. So you might have some obligate anaerobes that are able to survive and continue doing anaerobic respiration or maybe fermentation, depending on what is going on here, in order for them to survive. Okay. All right. So let's go ahead and do some review questions. So here is our review question. Which of the following fermentation methods can occur in animal skeletal muscles? So what do you think, what type of fermentation methods can occur in our cells? Is it A, mixed acid fermentation, B, alcohol fermentation, C, carbon dioxide fermentation, or D, lactic acid fermentation? Which one can occur in our cells? If you chose D, I would agree with you. So we can perform lactic acid fermentation. Okay, so this is, again, when we're trying to reach the 100-meter mark. We're at the 80-meter line. We have 20 meters left. We're out of oxygen, so we got to kick in to lactic acid fermentation to get us just enough energy to get us to the finish line. Okay, we cannot do alcohol fermentation, and if you think about it, that would not be a good idea, because then we would be drunk when we exercise, right? Like the crash course video suggested. So D is the correct answer. Okay, so that is it from our fermentation video. So by now you should be able to discuss the fundamental difference between anaerobic cellular respiration and fermentation. You should also be able to describe the type of fermentation that readily occurs in animal cells and the conditions that initiate that fermentation. Okay. So for our final video for chapter 5, we're going to get into the conditions to other metabolic pathways. So what we can use aside from glucose in order to make ATP.